In this fourth video, and yes, I know it keeps going on, but there's a lot going on, and this would normally take a few 1 hour lecture periods to go through, but we're going to try and put together some of these cellular defenses and see how they play a role in this process that's going on here. So this is where we start to see some of the stepwise or ordered fashion for the immune response to an invading pathogen or the particles created by that pathogen. And if we look at this process, we have to think about how do these white blood cells that we saw in the last video, these leukocytes, how do they recognize that you're actually in the presence of a pathogen? And this is where, unfortunately, we've got some more acronyms. We've got PRRs that are binding to PAMPs. And what we're looking at here is pathogen recognition receptors. That's the PRR that you're going to find on the outside of your leukocytes. Think of these as little hands, and each hand is a slightly different shape because it binds to different pathogen-associated molecular patterns out there, or PAMPs. So here we've got a receptor that binds to lipopeptides. Here, a receptor that is specifically made that binds to flagellin proteins or peptidoglycan. And if you think about this, when this pathogen recognition receptor on the cell binds to its ligand that came from the pathogen, that sends a signal to the nucleus of this cell that turns on certain genes, and that's going to cause this cell to ramp up its activity. If it's a phagocytic cell, it's going to start turning on some things to get ready to kill and digest the pathogen that it's eaten by the process of phagocytosis. You're also going to start to see an uptick in the production and secretion of these different kinds of cytokines, including our interferons that are out there. And so if we think about this, all of these different things that are out there can be used to recognize it, but also remember, if we look back at our complement proteins, they can opsonize things that are out there. And so effectively, they're going to coat things with these flags so that your cells can identify them as foreign objects that are out there. So when we look at this, the book talks a little bit about toll-like receptors, and this is just one group of these things. So if you wanted to, you could put another little thing over here that is going to basically function like these other ones that we see here. They bind, they recognize and bind to the pathogen associated molecules that are out there, like our flagellin or our peptidoglycan that's out there. And then when we look at this process and think about, okay, now you've got this cell that has been activated by binding to this thing right here. Well, what role do these phagocytic white blood cells, these macrophages that are distributed throughout your body, both in tissues and circulating, what are they going to do? And effectively, when we look at this process here, what are these phagocytic cells going to do? They are kind of like cops. They are going around into the tissue compartments. They're going around in the fluids in your body, and they are holding out their little hands to bind to these foreign materials that are out there. And if it's a phagocytic cell, their job is to eat it. And so they're going to ingest and then get rid of these foreign material that's out there. And we'll see in the next chapter, they're going to also help to recognize some of this information and give it to trigger parts of our immune system that are really specific for those particular pathogens. If we look at this process of phagocytosis, we can look at it as, well, first you start out with your phagocytic cell is going to migrate to the site of an injury that's out there because of a specific chemical that's given off there. So think chemotaxis. You are moving in response to a chemical that is being released by cells in the area. Now that you've moved there, you're going to ingest it and bring that pathogen in by phagocytosis here, and that is going to form what's known as the phagosome. So this is basically a vesicle or sac filled with your pathogen that's being brought in from the outside there. And now that you've ingested it and you've got this phagosome, you can fuse this stuff with a lysosome out there. So your vesicle that is full of these digestive enzymes that are out there. And when we fuse these guys together, you're effectively dumping your stomach contents onto that particular pathogen that's out there. So in this phagolysosome right there, you're going to start to destroy that foreign object by basically digesting it into little bits and pieces. And we're looking at something that's going to take place in minutes with all the different kinds of antimicrobial chemicals that you're going to be releasing there. Now that you're starting to digest it, we are going to completely destroy it. So we're going to release all of these different kinds of antimicrobial chemicals, including the stuff with halogens in it, like bleach or forms of bleach that have the oxidizers that are out there. We're also going to be dumping superoxides on it, singlet oxygen, free radicals that are there. We're going to be releasing all of these different digestive enzymes. You're going to pump protons in there, and you're going to make it more and more acidic. So you're dumping all of these things into this phagolysosome to basically destroy that particular particle right there. And then you are left over with some of this undigested waste product. And some of this will get saved, as we'll see when we look at antigen processing, and the rest of it is going to get excreted by the cell right there. And this is an important thing because those are chemotactic signals for other cells that are out there. And also this phagocytic cell is going to start releasing various cytokines to signal to other cells, hey, I have eaten something that's foreign. You guys need to come to this area, and that can activate things. You release interferon alpha and beta from these cells that can activate other cells in the surrounding neighborhood right there. So if we look at this process, two of our white blood cells that are granulocytes are going to be phagocytic. Your neutrophils are extremely important. Remember, most or the majority of white blood cells that you'll find in your bloodstream are neutrophils, and these guys actually will see an increase in numbers if you do a blood cell count early on in a bacterial infection. If you have a high number of neutrophils, that is actually a signal of a bacterial infection going on. If you've got what's known as eosinophilia, you've got an excess number of eosinophils floating around, that is a good indication that you've got a parasitic worm infection inside of you, or maybe you've got and some allergic response going on. But both of these guys are going to be able to do this process of phagocytosis. We've also got these mast cells that are floating around in your bloodstream, or the various histiocytes that we'll find distributed throughout your body that are also phagocytic in nature. And when we look at these guys, remember their job is to eat things and clear out the waste products that are produced as a result of your immune system response. So let's take a look at this process of your immune system response here and some of the steps that are going on. If you think about it, you develop an injury right here. You have a break in that first line of defense through this epidermal layer that allows a pathogen to get access into your body. The cells in the surrounding area right there are going to start releasing chemicals that are going to trigger other elements of your immune system there. And so when we look at this, this is spreading from the site of the injury, and it can spread throughout these extracellular spaces, but it can also start to work its way into your lymphatic system and eventually to your bloodstream, where it could go systemically throughout your body there. And so when we look at this process here, these chemicals, these chemokines are going to be released to activate various cells of your immune system so that they can start getting ready to fight off the infection that is going to occur there. When we look at this early event, I stole this figure from a different textbook that's out there, so you can see during this early process here, you're going to be releasing these various chemical mediators into the surrounding area. And one of them, or I should say one group of them, is what's known as vasodilators. And if you look at this thing here, you can see this element of your circulatory system is a smaller tube, and in response to these chemicals that are being released from these cells in the surrounding area there, you're going to have vasodilation. So this little capillary bed is going to start increasing in size, and that's going to allow some of the fluids in your blood, remember your blood plasma, is going to leak out and bathe this area in that surrounding region right there. Well, think about it. That blood plasma contains a whole bunch of protein elements. It's got things like your complement proteins. It's got defensins that are floating around. It's got these iron binding proteins. And now you've just increased the amount of those fluids in that local region right there. And so if we look at this, we can see these little guys right here, these chemicals that could be released in the area there. There's that histamine. If you remember, our basophils that are out there are carrying a lot of this chemical around. What does it do? Causes vasodilation that's out there. It's actually going to cause an increase in secretion of mucus. Well, that's what you want. You're going to have more and more of these chemicals in the area there. You've got leukotrienes that are basically long-term histamines that are out there to promote this inflammation that's going on in the region. You've got prostaglandins that are really local hormones that are going to help to promote that inflammation that's going on. And it's kind of interesting when you look at aspirin, it actually is going to inhibit certain prostaglandins from being made, but at the same time it does reduce platelet activity. So you might block the effects of what you're feeling there with the prostaglandins, but you're also going to lose the effects that they are actually doing to boost your immune system response there. When we look at the second step here, now that you've gone through this process of activation, you're going to have chemotaxis. You're going to have the attraction of cells that are traveling through your bloodstream to the site of the injury right there. So these leukocytes, think monocytes, those small cells that are floating around there, they're going to be able to do what's known as transendothelial migration. They're going to go across this layer of cells in that capillary bed right there. They will stick to the margin or the edge of this tube right here, and diapedesis, think diameter, you walk across there, and they are literally squeezing in between the epithelial cells, and they are moving to the site of that injury in response to, in this case, our complement protein that was activated by complement cascade in the region where that infection is occurring there. Now you go through your phagocytosis process, and we saw all the different steps there, and you're going to release those cytokines here that can cause even more chemotaxis there, and the cells are going to swarm the site of the infection there and clean up the area there and release more chemicals to attract even more cells there. And so if we look at this process here, these damaged cells and the macrophages that are eating things are releasing cytokines. They're activating complement in the area there, and we're going to get that vasodilation, or this increase in the amount of fluids that are going on there. And this is where we see the four classical sort of signs and symptoms of inflammation. You're going to have redness in the area because you've got an increased circulation and more of red blood cells are traveling to the area there as you get vasodilation in the injured tissues with the capillary beds there. You're going to see an increase in heat in the general area there because there is more blood going to the sign or to the site where you've got that increase in fluids. You're going to have swelling as a result of that. You're going to experience some pain because of the pressure of all those fluids, and you're going to have that edema occurring there as you get that swelling in response to the inflammation that's going on there. Some people also talk about this contribution of the loss of function that occurs as a result of inflammation. So now that you've got inflammation, what's the point? Why do we do this? And if we look at it, you're going to have acute inflammation that is quick, and it's going to end, and the tissue can be repaired there. And they've actually been able to demonstrate that some of these phagocytic cells actually work better at a slightly elevated temperature there. So that's great as long as it's quick. And if we look at this, you can see there are different categories of fever here. Your normal temperature is going to be a range right here. If you have a moderate fever, that's actually okay to have. But if you get above 104 degrees Fahrenheit, especially for an older person, that can actually be life-threatening. So if you have a chronic kind of inflammation going on that results in a fever, there you have a battle that is continuing to go on between the host and the pathogen. And the longer you have inflammation and fever, the more likely you are to get damage to the host. And that can result in some long-term effects that are out there. So if we look at this process here, your leukocytes that are out there are going to be releasing a variety of different chemicals known as pyrogens, or they're going to create a fire, and that's going to be responsible for you getting a fever. We normally set our body temperature from our hypothalamus right here, and so these endogenous pyrogens are being produced by your cells that are resulting in your hypothalamus basically resetting its temperature right there. There's also exogenous pyrogens, and remember our endotoxin with our lipopolysaccharide that's out there. So this could be a chemical that is being released by the pathogen that is actually causing your body to result in a fever or produce a fever as you reset your body temperature. And so the longer that happens, the more damage can occur to you as a result of that. The last sort of stuff we can look at is, okay, why do we do this? Well, it's a good thing because it tends to inhibit the growth of certain pathogens that are out there. It also is going to increase the ability of your immune cells and your ability to sequester that iron. And then when your fever breaks, you know, because you're going to sweat. Your body's going to do that response to try and cool down out there. And so when we look at a fever, it's really one of those things where it's a good thing as long as it goes away, but you have to decide when is it quick. And that's the problem with looking at this. The more experience you have, the less risks you're going to be taking potentially, especially when you're looking at things like people who have other things that are going on. They've got cardiovascular disease or they're experiencing some sort of respiratory ailment. We want the fever, but we don't want too much fever out there. So just to go back and think about, we've been looking at all of these different elements, your physical and chemical barriers on the exterior of your body to prevent things from getting in, and then the underlying mechanisms like phagocytosis, all the elements in your bloodstream that are out there, and then this process of inflammation. All of these things are in play since before you're born, but remember, they also interact with the other side of your immune system that we take a look at in our final chapter, where we look at the adaptive immune response that you actually build up throughout your lifetime that is going to be more specific and help to destroy the specific pathogen. And if it comes back, you remember it so that you can destroy it again and again and again.